Thumbs up?
Yep, that's good.
All right, excellent. So first of all, I want to thank David and all the attendees really for the opportunity to speak at this meeting. I have very fond memories of the first XLP symposium and a couple after that, and I fear we've been away for a little while, but it's so nice to be back, even if I can't come to London, so thanks again. So today I'm going to focus on apoptosis as the general theme, and I fear as my lab's drifted a little bit away from XLP, this will be largely some review, but hopefully I'll take you through some of the work we've done over the years to convince you we've learned a lot about T-cell apoptosis by studying XLP1, and then it's hopefully come back around to talk about some important consequences for the specific disease itself, XLP1. So as most of you know, of course, this is a nice histograph from one of the original reports of a family with what was later determined to be XLP. And the point I want to illustrate here is just the lymphoproliferation, right? These are all CD8 effector T cells that are invading the liver. And so the first thing I want to do is just remind everyone, of course, that the L stands for lymphoproliferation, right? Lymphoproliferative disease. So in my view, XLP1 really is fundamentally a disorder of perturbed immune homeostasis. And we know the most common and certainly life-threatening clinical feature of XLP1 is this EBV-induced infectious mononucleosis and HLH. So obviously excessive accumulation of CD8 positive effector T cells is a major contributor to that immunopathology, even though we know from the work of folks like Stu Tang and many others that there seems to be a specific impairment of cytotoxicity against EBV-infected B cell targets. So over the years, through the work of numerous groups, including many on this call, we know the SAP and SLAM receptor system plays several important roles in managing both innate and adaptive immunity. So we know it's important for providing B cell help through CD4 T cells, and impairments there might explain this gamma globulinemia. We know CD8 positive and NK cell mediated cytotoxicity, particularly against B cells, is defective. And as I'll focus on today, we also know that CD8 T cell apoptosis seems to be perturbed in the absence of SAP. So this is a slide I use in a lot of talks, and this is just to sort of ground everyone in the idea that apoptosis is pretty important for shaping both the magnitude and potency of a T cell response. And so as T cells clonally expand, my lab in particular gets interested right about here when we talk about the peak of the immune response and two major apoptosis pathways that are responsible for not only controlling the magnitude of this response, but then eventually contraction of that response. So re-stimulation-induced cell death, which is the focus of my talk today, is this self-regulatory or what we call propriocytal death pathway, whereby a proportion of T cells, if they're re-engaged through the T cell receptor by antigen, undergo apoptosis. And we think this pathway, and we know, frankly, by studying XLP1, is particularly important for constraining excessive T cell expansion, basically limiting the number of effector T cells that are eventually made. Cytokine withdrawal-induced death is arguably the more important pathway for contraction and culling of effector T cells after a pathogen is cleared and the cells stop making interleukin-2. I won't talk a lot about that today, except to remind you that we know lymphoproliferative disease has been linked to gene defects in things like FAS-induced apoptosis in the case of ALPS, as well as cytokine withdrawal-induced death in the case of RAS-associated leukoproliferative disease. So back when I was a postdoc at the NIH with Mike Leonardo, we asked a fairly simple question about XLP1 patients in that could the CD8 T cell lymphoproliferation that is a hallmark of this disease be tied to a specific apoptosis defect? And the bottom line answer is yes, and I apologize, it's hard to believe this data is well over 10 years old now, but in collaboration with Rebecca Marsh at Cincinnati Children's Hospital, we were able to show that indeed XLP patients, shown in the blue, have a specific apoptosis defect in this RICD pathway, and that when you re-stimulate effector T cells in vitro with an anti-TCR antibody, you can see that they die a lot less readily. And it seemed to be specific to that pathway in that if you directly ligated fast or you treated with staurosporin, UV irradiation, or even cytokine withdrawal, the cells died just fine. So this RICD path defect seemed to be specific to XLP1. We could tie it through knockdown experiments, et cetera, to a specific loss in SAP expression. And we think that was a fairly important finding in understanding some of the mechanism behind EBV-induced mononucleosis and HLH. In that we know from work from Stu Tanguy and Pam Schwartzberg and others that the priming of CD8 T cells by professional antigen-presenting cells like dendritic cells seems to operate just fine, and that you get EBV-specific CTLs generated. But at the point which you're asking those CTLs to kill EBV-infected B cell targets, there's a defect in conjugation with those B cells. And indeed, instead of, in a normal situation, removing those B cells by cytotoxic killing, and the RICD pathway that basically ensures we don't get too many CD8 effector T cells, unlike the normal TCL homeostasis here, we get this sort of failure to control the EBV-infected B cells, as well as a pileup of effector CD8 T cells that can't regulate themselves through RICD, and that gives rise to this FIM HLH phenotype. So this is just illustrated a different way, back to the diagram I showed you earlier, in that we think that XLP1 in particular exposes the physiological importance of this pathway. So when I first learned about this as a student, it never made any sense to me why you'd want to kill T cells at the exact moment you need them. But the point is that if you don't have this constraint in place, instead of controlling the number of effector CD8 T cells properly, you can get an uncontrolled expansion. And in XLP1 patients, in exposure to most infections, you might get a transient lymphadenopathy, splenomegaly, accumulation of CD8 effector T cells, but the cytokine withdrawal pathway operates normally once that pathogen is cleared. But because they are uniquely susceptible to EBV infection and that they cannot clear it properly, this RICD defect really comes into play, and you get this extreme accumulation of CD8 effector T cells. So the next question, of course, is why? Why is there a defect in RICD in the absence of SAP? And this slide is meant to summarize quite a bit of work we've done since I started my own lab at USU back in 2011 to kind of unveil some of the mechanism behind this pathway. So we knew from our first study that when we lost SAP expression, that the overall signaling strength through the T cell receptor, which is very important for driving cells beyond the threshold to commit to apoptosis, was weaker in that you got a globally reduced TCR signal strength, and that led to a poor induction of molecules like Fas ligand and BIM, these pro-apoptotic molecules that execute RICD. And then we've known from subsequent work on the molecular level that we think SAP facilitates RICD through a number of ways. First and foremost, it seems to operate through a specific SLAM family receptor we know as NTBA or SLAMF6. And it does that by not only displacing phosphatases like SHIP1, but also recruiting and activating more proximal kinases, particularly LCK, that seems to amplify the proximal signaling pathway. And then in a wonderful collaboration with Andrea Graziani and Gianluca Baldonzi at Novara, we also know that SAP seems to be important for blocking DGK alpha activity and sustaining levels of this important secondary messenger in the TCR pathway known as diacylglycerol. So we showed, and I'll show a little data here just to review, that just by inhibiting this one enzyme, DGK alpha, which is more active in XLP1 patients, you could not only restore the RICD pathway in vitro, but in vivo, you could actually show that this had a beneficial effect by reducing what looks a lot like EBV-induced FIM in a mouse model. And I'll just take you through that briefly. So we were able to show in vitro that whether you knocked down DGK alpha using siRNA or you blocked it with a pharmacological inhibitor, you could partially restore RICD sensitivity in XLP patient cells. These are CD8 effectors. And of course, this was an apoptotic pathway, and you can see a nice restoration of the Nexin 5 staining. This is an early hallmark of apoptotic cells when you had the DGK alpha inhibitor on board for XLP patients. I won't have time to go through all the data. This is long published, but we were able to show again in collaboration with Andrea's lab, that it seems to work a couple different ways. So by blocking DGK alpha in SAP-deficient T cells, we could basically restore levels of diacylglycerol that facilitated immune synapse formation and the recruitment of these important signaling molecules, PKC theta and RAS group 1. And that seemed to be important for ensuring you had proper IL-2 signaling, which is critical for maintaining RICD sensitivity. But it would also drive this restoration of RICD itself specifically by inducing these novel proapoptotic molecules, NOR1 and NOR77, which we know antagonize the activity of an anti-apoptotic protein called BCL2 through phosphorylation by this ribosomal S6 kinase. So I know that's a mouthful, but suffice to say, we were able to work out some of the molecular details here, and it appears that DGK alpha itself was controlling specific parts of the apoptotic program that are responsible for RICD. More importantly, with Kim Nichols and Rupali Das at St. Jude's, we were able to demonstrate efficacy in vivo using the LCMV model. So I'm sure most folks on this call know LCMV is an excellent mouse viral infection model for examining CD8 T cell responses. And so what Rupali did was to basically inoculate either wild type or SAP deficient mice with LCMV, allow the CD8 T cell response to ramp up over 4 days, and then treat those mice twice a day with the DGK alpha inhibitor. And what was remarkable is that you can see that with LCMV infection, you get splenomegaly even in wild type mice. When you added the DGK alpha inhibitor, you got a slight reduction in the number of cells. But more importantly, if you look specifically at the LCMV specific T cells, the proportion of those cells didn't change a whole lot between wild type and SAP deficient cells, but the number of them did, which suggested to us that we in fact were restoring apoptosis sensitivity. And again, we're only introducing this inhibitor well after the CD8 T cell response is primed in these mice. And then if you look at a hallmark of what looks a lot like HLH or EBV-induced FIM, again in the surrogate model, the formation of these foci, these inflammatory foci in the liver. Remarkably, in the SAP-deficient mice, you could see the number of these foci reduced, and more importantly, the size of them, just by inhibiting DGK alpha with this single pharmacological inhibitor. So that was really exciting to us, that you could basically correct some of this important hyperinflammatory state, this pathology we see in XLP1 patients, just by inhibiting this single enzyme. So we always come back to the original question, which I think still remains a bit unresolved, which is how SAP exactly inhibits this enzyme. Gianluca showed years ago that in fact it does in a really nice JI paper, but the mechanism has remained somewhat obscure. Elisa Rufo who spent some time in my lab, she was a graduate student with Andrea, did work out some of this pathway, which remains unpublished, in that we know SAP interacts with several other proteins that are involved in cytoskeletal remodeling and actin polymerization. And she was able to show that indeed, if you knock down any of these pathways in sort of the actin polymerization pathway, as you might expect, you get a reduction in RICD. But if you inhibit DGK alpha, it generally restored that. And it might have something to do with beta-1 integrin signaling in particular, in that if you knock down, if you knock down beta-1 integrin, again, you get a nice, pretty profound reduction in RICD sensitivity. But by inhibiting DGK alpha, you could largely restore that. So this is very much still unresolved. And I think I'm really looking forward to Dr. Graziano's talk, because I think he might shed some light on this today. But suffice to say, there's more work to be done in exactly how this pathway works. So from that point, we sort of asked, we put SAP and NTBA squarely in the RICD pathway, which by that time, no one had known SLAM receptors were particularly involved in apoptosis. But we started asking questions about whether SAP and NTBA sort of serve as normal rheostats or controls of RICD sensitivity in normal, healthy human donors, which show quite a bit of variability in their sensitivity to this pathway. And I'll just take you through some work we've published over the years on that question. So if you simply look in sort of conventional CD8 T cells, the answer, I think, is squarely no. in that if you look at SAP expression over a number of donors, I'm just showing you a snapshot here of dozens we've looked at. There's really no correlation just comparing the amount of SAP expression in their CD8 T cells to their sort of maximum RICD sensitivity. Occasionally we'll see a donor with quite low SAP expression that also has very low RICD, and so perhaps there's some kind of threshold effect, but by and large there's really no correlation there. Certainly not compared to others we've shown like glycolytic flux, and this was Sasha Larson, who worked on the DG Alpha paper, was able to show in a separate study that certainly the more glycolytic CD8 T cells seem to be, that correlated beautifully with their sensitivity to this DEF pathway. So SAP is not really serving as a rheostat in conventional CD8 T cells, but remarkably in regulatory T cells and CD4 Tregs, the answer is squarely yes. And this is work from Kelsey Voss and Gil Katz, who also worked on some of the XLP1 work as well. And this was based on the premise that had already been published that Tregs are quite resistant to RICD, but it wasn't really clear why. And so we were able to recapitulate that by simply sorting conventional T cells from Tregs and asking, are they sensitive to RICD? And Tregs clearly are not. But remarkably, if you look at those Tregs, they have very low expression of SAP. And as I'll show you in a minute, that seems to be important for their RICD resistance. And it correlated inversely with their expression of FOXP3, which of course is the master regulator of Treg function. So if you look at the actual promoter for the SAP gene, you can see FOXP3 clearly binds to it in regulatory T cells, but not conventional T cells. And in fact, if you look at the promoter for the SAP gene, you can find lots of FOXP3 binding sites. We could show in a reporter assay where we hook up the SAP promoter that FOXP3 certainly suppresses the activity of that promoter. If you introduce a DNA binding domain mutant, it's abrogated. And then more importantly, we could show that SAP, the repression of SAP is both necessary and sufficient for resistance to RICD and Tregs, and that if you knock down FOXP3, you can restore their RICD sensitivity, and that requires SAP if you do a co-knockdown. But more importantly, if you simply overexpress SAP through a lentivirus, a different promoter, you can restore RICD sensitivity in Tregs. So clearly, suppression of SAP is important for maintaining that population by repressing their sensitivity to RICD. So Kelsey went on to show in conventional T cells that is not the same effect for FOXP3 in human conventional CD4s, and that we knew that FOXP3 was transiently upregulated in conventional human T cells after initial antigen stimulation. And she hypothesized that maybe that was also contributing to this early resistance to RICD we see over the course of the T cell response. And in fact, that was true. So if you knock down FOXP3, both in CD4s and CD8s, you can see a nice increase in RICD sensitivity. But interestingly enough, that seems to have nothing to do with SAP, in that we see no increase in SAP expression if you knock down FOXP3 in those conventional T cells. More importantly, if you simply knock down SAP, much like we did in sort of the late stage effector T cells at this point, in early expanding effector T cells, knockdown of SAP really doesn't do anything to change RICD sensitivity, which is interesting and we're following up on. It suggests that SAP is not playing an important role in this early phase of the response, and that there's this temporal dependence on the SAP NTBA signaling pathway for RICD control. So that's not to say that the question's completely answered. So it could be very much context dependent, and I'll just point to a nice study by this group that published a few years ago that if you look in TB patients, for example, at their CD4 T cell response specifically to Mycobacterium tuberculosis, you can see that the patients that were quote unquote low responders had higher expression of SAP, but that they were actually more resistant to RICD, and it seemed to have something to do with this preferential recruitment of FIM-T, which we showed years ago is not happening in normal TCR restimulation. Normally SAP recruits LCK in that case, but perhaps because the SAP is higher, there's some preferential recruitment of FIM-T, and they were able to show that indeed this interfered with normal fast ligand upregulation and apoptosis through the RICD pathway. So it could be context dependent, and points to the fact that there could be a lot more work to be done here to understand how this works. So just to go briefly over what I've told you today, so to summarize, for XLP1 patients, we know that SAP deficiency is clearly tied to RICD resistance. And we think this is an important phenomenon that explains the exacerbation of overexpansion of CD8 effector T cells in that disease, and part of the immunopathology that drives infectious mononucleosis in HLH in response to EBV. We knew by further defining these signaling mechanisms that we'd likely find some therapeutic targets, and DGK alpha inhibition is a very promising strategy. And I think you'll hear from Suresh in the next talk some follow-up on that particular study. And then I don't know about you guys, but I've been sucked into the COVID-19 vortex, and we're doing some research. And so it made me think a little bit about what the potential consequences would be for XLP1. And I would say the concern would be warranted here in that we know in severe COVID-19 patients, you see a pretty massive expansion of CD8 T cells early that then drops off, and you see lymphopenia that seems to be associated with a lot of cell death, a lot of exhaustion. I would worry that because RICD is not acting properly in XLP1 patients, that potentially those patients would be more prone to CD8 T cell immunopathology in that disease, particularly in kids with MIS-C. To my knowledge, there's no reports of that quite yet in the literature, but something to look out for. And then for others outside of XLP, we know from our work that SAP and NTBA and FOXP3 don't seem to be reliable predictors of RICD sensitivity, but it depends on the context. And that if you look in Tregs, certainly FOXP3 repression of SAP plays an important role in protecting Tregs from RICD. And in fact, subsequent work has suggested that if you, in a rampant T cell response like HLH, you actually lose IL-2 and FOXP3 in those Tregs, and perhaps they're lost because RICD is now restored in that population. And then we also know through some nice work lately that NTBA, SLAMF6, seems to be potentially a marker of exhaustion in CD8 T cells. And so by blocking that pathway, it seems to be pretty useful in mouse tumor models for enhancing the CD8 T cell response. And we're starting to wonder whether that might be because you're actually decreasing RICD sensitivity in that context, which could be beneficial, of course, for driving a good anti-tumor T cell response. So with that, I think I'm still on time. I just want to thank everybody involved, both from my lab and then our wonderful collaborators at NIH, over in Italy, St. Jude's, and Cincinnati Children's. And I'm happy to take questions if time permits.
Thanks, Andrew. There wasn't any questions that came in on the Q&A. Are there any questions for Andrew now?
I see one from Troy. Yeah. So I think I will defer to Suresh, our next speaker, on where we stand on the DGK alpha inhibitors, because they've done some lovely work over in Italy to follow up on that.
Okay, that sounds always sounds good when it looks like the agenda might actually make sense.
If I've done nothing else, if I've set up better talks in front of me, then I've done something right. So, but thanks.
Thanks very much, Andrew. I'm going to reclaim the host.